clarity clinical solutions.
Data cleaning is the systematic process of reviewing clinical trial data to identify and correct errors, inconsistencies, and omissions. In clinical research, the quality of conclusions depends entirely on the quality of the underlying data. Even small errors can lead to incorrect findings, potentially harming patients or derailing drug approval. Data cleaning ensures that the information collected from study sites is accurate, complete, and reliable before it is analyzed. This module explores the principles, processes, and best practices that make data cleaning a cornerstone of clinical data integrity. Clean data directly impacts patient safety and regulatory decision-making. Inaccurate data can mask safety signals, exaggerate treatment effects, or introduce bias that undermines trial conclusions. Regulatory agencies like the FDA and EMA require documented evidence of data quality control procedures. Poor data quality is a common finding during regulatory inspections and can lead to clinical holds or rejection of marketing applications. Beyond compliance, clean data enables faster study closeout, more efficient statistical analysis, and greater confidence in the results that ultimately guide medical practice. Clinical trial data can suffer from many types of errors. Missing data occurs when required fields are left blank or visits are incomplete. Out-of-range values may indicate incorrect units, transcription errors, or equipment malfunction. Inconsistent data appears when two related fields contradict each other, such as a male patient with a pregnancy test result. Protocol deviations may be recorded incorrectly or not at all. Duplicate records, incorrect dates, and illogical sequences are also frequent issues. Each type of error requires a specific cleaning approach and documentation. Data cleaning follows a structured workflow that begins when the first patient data enters the database. Initial checks include automated validation rules that flag missing values, range violations, and logic errors. Following automated checks, manual review by data managers identifies more subtle issues. Queries are generated for sites to resolve discrepancies. After corrections are entered, the system revalidates the data. This cycle repeats throughout the study until database lock, when all queries are resolved and the data is declared clean and ready for analysis. Edit checks are programmed rules built into the electronic data capture system that automatically flag potential errors during data entry. Range checks verify that values fall within expected parameters, such as a heart rate between 40 and 200 beats per minute. Logic checks confirm relationships between fields, ensuring a date of birth precedes the date of informed consent. Consistency checks cross-reference related measurements. Missing data checks identify incomplete forms. These automated rules provide real-time feedback to site staff and reduce the burden of manual review. While automated checks catch many errors, human review remains essential. Data managers and clinical research associates review patient records holistically, looking for patterns that automated rules might miss. Manual review identifies implausible clinical narratives, unusual medication patterns, or subtle protocol violations. Reviewers examine source documents against case report form entries to verify accuracy. This process requires clinical knowledge to distinguish legitimate unusual values from true errors. Manual review is particularly important for adverse event coding, medical history interpretation, and narrative data. Source data verification, or SDV, is the process of comparing data entered into the case report form against original source documents. Source documents include medical records, laboratory reports, imaging results, and patient diaries. SDV is performed by clinical research associates during site monitoring visits. The extent of SDV is typically determined by a risk-based monitoring plan. Critical data points such as informed consent, eligibility criteria, and primary endpoint measurements receive the highest priority. SDV ensures that the data in the clinical database accurately reflects what actually occurred at the study site. Modern clinical trials rely on sophisticated electronic data capture systems and data management platforms. Tools like Metadata Rave, Viva Vault, and RedCap provide built-in validation, query management, and audit trails. Clinical data management systems support configurable edit checks, discrepancy tracking, and automated workflows. Statistical programming tools like SAS are used for advanced data validation and discrepancy detection. These systems maintain a complete audit trail of all data changes, ensuring transparency and regulatory compliance. The choice of tools depends on study complexity, sponsor requirements, and budget. Clinical data managers are responsible for the overall quality of trial data. They develop the data management plan that outlines cleaning procedures, edit check specifications, and quality control measures. Data managers coordinate the review process, track query resolution, and ensure timely database updates. They work closely with biostatisticians to prepare datasets for analysis and with clinical teams to resolve medical discrepancies. Data managers also oversee database testing, user acceptance testing, and the creation of annotated case report forms. Their expertise ensures that data is clean, complete, and audit-ready. When data discrepancies are identified, formal queries are generated and sent to the study site. Each query clearly describes the issue and requests clarification or correction. Queries are tracked through a lifecycle from creation through resolution and closure. Sites are expected to respond promptly with supporting documentation. All queries and responses are maintained in an audit trail for regulatory review. Data managers review responses to ensure they adequately address the discrepancy. Timely query resolution is critical for maintaining study timelines and achieving database lock on schedule. Data cleaning includes the important step of medical coding, where adverse events, concomitant medications, and medical history terms are standardized using established dictionaries. MedDRA, or the Medical Dictionary for Regulatory Activities, is used for adverse events. WHO Drug Global is the standard for medications. Coding converts investigator verbatim terms into standardized preferred terms, enabling consistent analysis across studies. Data managers review uncoded terms, verify auto-coding suggestions, and manually code exceptions. Accurate coding is essential for signal detection, safety analysis, and regulatory submissions. Database lock is the milestone when the clinical database is declared complete and accurate, and all modifications are permanently closed. Before lock, a rigorous checklist is completed. All queries must be resolved, all data discrepancies addressed, and all external data reconciled. The data manager certifies that cleaning procedures were followed. After lock, no further changes are permitted without a formal unlocking request, which is rare. The locked database is handed to biostatisticians for final analysis. Database lock represents the culmination of all data cleaning efforts throughout the trial. Regulatory inspectors carefully examine data cleaning records during inspections. They review audit trails to verify that all data changes are documented with the original value, the corrected value, the date, the user, and the reason for change. Inspectors assess whether cleaning procedures were followed as specified in the data management plan. They check for unexplained delays in query resolution and patterns of repeated errors. A robust audit trail demonstrating thorough data cleaning is essential for passing regulatory inspection. Any gaps in data cleaning documentation can raise serious concerns about data integrity. Data cleaning is a vital process that ensures clinical trial data is accurate, complete, and reliable for analysis. It combines automated edit checks with careful human review and systematic query management. The process runs continuously throughout the study, culminating in database lock. Clean data protects patient safety, supports regulatory approval, and enables confident clinical decision-making. Every member of the clinical research team plays a role in data quality, from site coordinators entering data to data managers overseeing the validation process. Committing to rigorous data cleaning is committing to scientific integrity.